All right, so what a great day. We are going to our afternoon keynote speaker, and this is Lorenz Studer. And I was very happy that he agreed to fly here and give us this keynote lecture on his work. So I got to meet Lorenz a while ago when I was still a postdoc, actually. And I was very surprised by how approachable and how nice he was. Actually, I mean, we still hear that good things about him. Yesterday, Elena Catanel just mentioned that. I mean, his ability to aggregate people to work together on something. And this is probably something that he's going to be talking about. So he's from Switzerland. He got his MD, Ph.D. from the Bern University. And then he did his postdoc at the NIH with Ron Mackay, who's other big name on stem cell biology. And really at the Switzerland, he was already involved on these trials for Parkinson's disorder. And I think that's what he got the idea that develop new stem cells models for that. But what really strikes me was all his work on pluripotent stem cells and trying to differentiate these cells in different subtype of neurons. So virtually, if you go in any stem cell meeting that is going on all over the world, you're going to see some reference to his work because his protocols is really amazing and really works. So we all are very glad that his lab has been troubleshooting lots of these small details to make more robust protocols. I think he would be talking about his work on trying to move into a clinical trial with these iPS-derived dopaminergic neurons. And please welcome to Dr. Lorenz. Yes, here.
Okay, great. So thanks so much, Alison and Luke, for the invitation. It's a real pleasure to be here and to close this exciting meeting. Now what I want to do is to, as the title implies, talk a little bit about both CNS and PNS derivatives and how you want to use them, particularly in the context of regenerative medicine. And that comes then really to the main tool that we've used and many other people now using routinely, which is the tool of differentiating neural cells using dual SMAD inhibition. And so this is a protocol where you can quite conveniently use two small molecules to basically convert pluripotent cells into neural cells. But what's maybe less appreciated is not just CNS neural cells. With just some small tweaks, you can actually basically make all the major ectodermal lineages, which includes also the neurocrest, the sensory placode, and the epidermis, which gives rise, for example, to the skin or to salivary glands and so on. And that leads then to the fact that we have now kind of an arsenal of different cell types. In my lab alone, we can make more than 40 different cell types routinely. And I just list here some of those that we think are particularly interesting to be used in the context of disease, either for disease modeling or for the ones here marked in red. These are all cell types where we've already done some proof of concept using those cells also in the context of regenerative medicine for cell therapy. For two of those, actually, I'm going to tell you a little bit more in detail where we stand on applying those cells in the context of cell therapy. But where I want to start off is actually an example of disease modeling. In fact, it's the example of disease modeling, which is the oldest one in our lab, one of the first examples of how to use iPS cells, which is familial dysautonomia. So this is a rare disorder, but quite fascinating because it's caused by a single point mutation, and it's the same point mutation in 99.5% of all the patients. And the gene that's affected is called IKB-CAP, and the puzzle kind of the disease is that this gene is part, or at least that's one of the main functions, part of the transcription elongation complex. So if you think about that, it elongates mRNA transcripts, has very fundamental roles, but the disease is actually quite specific. It affects the autonomic nervous system, which is part of the peripheral nervous system. So how come And so that was really the part many years back where we started using iPS cells to address that. And I'm not going to go into details because that's published a long time ago. But what he could show is that part of the reason why it affects the peripheral nervous system has to do with the fact that the point mutation causes splicing defect. It makes 2 mutants of the IKB cap gene, shown here, and only the wild-type form actually makes protein. And it turns out if you now make different lineages, you can show that only in the peripheral nervous system you have a very severe form of this missplicing event, and therefore very little of the normal protein left. And that could then explain how, for example, you have problems making autonomic neurons or other peripheral nervous system defects, such as migration of those cells. This disease was a good model also to test basically the potential of iPS systems for drug screens. Initially already we tested one of the compounds that was suggested to be of potential use in the disease, which is called kinetin. And this compound actually partially reverses the splicing defect and has since gone into the clinical trial. We also used the same system to then do actual drug screening, a library of about 7,000 compounds, and found other compounds that seem to be even more potent than kinetin, and which we hope some of those actually might move to the clinic as well. Now, what I want to show you here is kind of, again, the evolution of nearly by now 10, we had just actually celebrated 10 years of iPS research, kind of the evolution of where we are now with the same disease, still working on it, at least to some extent. And it's another question, which actually, we just heard from Lee a little bit of a version of that, which is the question, even though in this case the patients actually have the exact same point mutation, how come that there is quite a bit of variations in the disease symptoms? Some patients are much more severe, some are much more mild. How come? And so we were very interested in that to see maybe the very simple explanation could be, we just heard about levels of SMA, maybe it's levels of IKB protein or splicing. That was the difference between the different tissues. You had different levels of IKB. So we looked at that and actually found that in this is iPS-derived neurocrest cells, mild versus severe disease, there was actually no difference. So that really seemed to not explain it, but what was quite curious, if you went back to the disease phenotypes, we found that if you look, for example, at the migration phenotype, which is quite dramatic in the severe versus control, the mild cells actually didn't have that phenotype at all. When we looked at autonomic neuron generation, same thing. So the mild cells actually seem to have not a problem at all to make autonomic neurons versus severe cells. So the severe cells clearly had a very severe phenotype, and we want to make sure that they are fine otherwise. So we had many iPS lines from severe patients and subjected them again to a variety of differentiation protocols, for example, cortical differentiation, endoderm, or cardiac, and actually none of those we found a difference. So this again speaks to the fact that this is a quite specific autonomic disease in this context, and no difference between mild and severe. We also use CRISPR-Cas technology to then fix the mutation in the severe cells to see can you actually rescue the phenotype, and gratifyingly you can completely rescue, in this case, the autonomic neuron generation phenotype, which again suggests this is completely dependent on this mutation. But again, this doesn't teach us now anything about why the situation is such in the mild patients, and the mild patients then in our assays actually don't show any disease at all. So how is that possible, and why do they have even disease? So there we then kind of had the hypothesis, maybe in this case it's not really the problem of making those autonomic cells, but maybe in that case it's a problem of maintaining them. And in fact, what is here with some peripheral neurons shown here is that when you, under these conditions, make peripheral neurons, they can be maintained quite stably in a dish. But in both mild or severe patients, you see a dramatic loss, actually cell death of those neurons. So therefore, the simple hypothesis is that what we seem to have is that in the case of a severe disease, you really have a neurodevelopmental disease that's quite severe. But you don't really have that in a mild form. In contrast, both diseases seem to show a degenerative aspect, and in a way that matches, again, the disease where the patients get progressively worse throughout life. Now, from a translational perspective, it was important to see in patients that have both mild or severe disease, would our compounds that we've previously discovered, would they still work for this phenotype here? Because obviously the patients, they already went through the developmental phase, and we wanted to see that they work, and again, gratifyingly, actually they work quite well, giving further rationale that these drugs probably should be further tested in patients. Now the puzzle, again, that we still haven't obviously completely resolved, and I'm not going to resolve that today, is the exact mechanism. Now we think, again, it's not really due to just simple splicing difference, as I alluded to before. Now we did a small molecule screen where we actually tried to see, can you find compounds that make severe cells more behave like mild cells? And we found some compound that seems to work in a way by influencing actually the extracellular matrix conditions. And that was quite interesting because in parallel, we also did exome sequencing on patients, both mild and severe, and obviously compared that to many, many control exomes. And what we found is that if you do that you have a number of genes shown here that are basically unique, mutated in those FD patients, and at least 3 of those specifically just in those with a severe disease. And the one that seemed to have the best score is lamin B4, which again is obviously affecting extracellular matrix quite directly. And it's a gene that we haven't really causally proven to be the cause of mild versus severe, but it's interesting because it's most highly expressed in those peripheral neurons. So again, at this stage, we are at the stage in IPS modeling of this disease where we went through initial fact that we can model the disease versus no disease, to using that for developing drug screens and new drug candidates, to actually now being at the stage where we can go more towards truly personalized medicine and try to understand what really is different between individual patients and hopefully eventually treat those differences. Now, the second example of a peripheral neuron disease that I want to talk about a little bit is the enteric nervous system. It turns out this is actually the largest part of your peripheral nervous system, which is the enteric nervous system. You have so many enteric neurons that they actually supersede the number of neurons that you have, for example, in your spinal cord. And it's also a very diverse population of neurons. There are at least a dozen of different neurotransmitters that are found in the enteric nervous system. Now, it's a classic example of an autonomic nervous system because you can do these crazy experiments where you literally, I didn't do that actually, there were scientists in the 50s and 60s that did them, but you can literally behead an animal, cut off the head, and look what happens to the colon. And the colon actually, as long as it has enough nutrition, it perfectly secretes, it perfectly has peristaltic movement. So you don't really need CNS input for that to function. It's clearly important for many diseases as shown here, some genetic disorders, but also many common disorders such as irritable colon or gastroparesis that many people suffer from that are very dependent on that. And furthermore, it's often affected as a side effect of certain drugs, such as antidepressive drugs. When you take Prozac, typically you get constipation because it also affects basically on the serotonergic system in your gut. And so the disease that we initially wanted to study was Hirschsprung disease, which is the most common genetic form of an enteric nervous system disease. And it's basically caused by the incomplete migration of the enteric nervous system during neurocrest delamination and colonization of the gut. Now, the most common genetic forms of this disease are mutations in RET or EDNRB gene, and the only way you can treat the disease currently is really by actual resection of the part where the ganglions are missing. If you don't do that, what happens is you get basically blockage of that area, and you get basically eventually a bursting of the gut, and obviously sepsis involved, and the child dies. Now, we wanted actually to use these cells in 2 ways, both in a disease modeling way and also in a cell therapy way. And for disease modeling, we decided in this case not to use iPS cells, but just simply introduce the various mutations using CRISPR-Cas9, and then basically use those, study those cells in the enteric nervous system. Now, it took us quite a while to actually make specifically enteric neurocrest cells, and it turns out that similar to the central nervous system, where you can make genetically neural cells, you can make genetically neurocrest cells, and by default, similar to the CNS, they are the most anterior portion of the neurocrest, the cranial neurocrest. If you want to make more posterior versions of the neurocrest, such as the vagal region, that gives rise to the enteric nervous system, you need to caudalize the neurocrest precursors, and you can do that, for example, with RA, where you see more than 1,000-fold induction of HOX2 to 5, but not HOX9, which is a bit like the zip code that tells you that you are at the level of the vagal neurocrest, but not, for example, of the sacral neurocrest or any other regions. Once you have done that, you can then take purified neurocrest precursors, characterize them for many, many different genes, and show that you get near pure population of cells expressing, again, markers of enteric neurocrest. And we've did a lot of profiling studies to now compare for the first time the various lineages, biased neurocrest populations. But the really interesting part was obviously, are they functionally different, not just molecularly? And one very simple assay you can use is to inject those cells into a chick embryo. In this case, inject them into the dorsal part of a chick embryo and see where the cells manages to migrate ventrally into the developing gut. And what you see here is indeed human cells, but they took up shop in the chick gut. And this is quite specific because you can take either cranial neurocrest or melanocyte biased neurocrest where we don't see such a response. Now the key feature, I think, for me at least, to feel comfortable that we indeed have enteric neurocrest was the fact that we can recreate the quite enormous diversity of neurons generated from those lineages. And so what we did here is first we purified them for SOX10, so we know that we work only with peripheral neurocrest precursors. And then we show that we can get the various neuronal lineages, for example, the classic motor neuron-like lineages, the NOS and the CAT neurons, but many other lineages as well, in a certain stoichiometric fashion. You can also show that these cells know where to home. This is an assay that's an organoid assay, which was done together with Tracy Grickscheid. And what you can do there is basically you have primary intestinal epithelial cells, and you mix in your human ES-derived enteric cells, and you see where they are localized. So these basic organoid-like structures, they reform the epithelium, and you find indeed that the human cells are either submucosal or in the myenteric plexus located. You can also test their function, in this case by putting in an optogenetic switch into the cells, which allows you then selectively only to stimulate those human cells. And if you do that, for example, here in combination with human ES-derived smooth muscle, you can appreciate this very slow, nearly peristaltic-like waves you can induce, which is again quite different from what I'm sure all of you have seen for cardiac beating or muscle twitching. So again, suggesting that this is triggerable, and in this case, smooth muscle actually doesn't spontaneously contract, as shown here, for a period of 80 seconds, but only once you stimulate the human cells. But the key feature really of those cells, or the most remarkable feature, was their ability to actually migrate not only in a chick embryo, but in an adult gut. And so what we did here is we injected them into the cecum, here actually first in the neonatal stage. So this is the part here most proximal of the colon. And then basically came back either after 1 hour, 2 weeks, or 4 weeks. And what you can see is that these cells managed to migrate from the injection site, and we actually put them there with a block of matrixol to make sure that they don't escape and just diffuse. So after 1 hour, again, only one bolus of those cells. But after 4 weeks, they actually colonized pretty much the whole length of the colon, which is nearly about 3 inches of length. Now the cells, again, when you then analyze those mice, you find that they are localized both in the myenteric plexus and in the submucosal plexus, and you find quite a diversity of both neuronal lineages, just as, for example, serotonergic neurons coming from human origin, or even enteric glia that was derived from those grafted cells. And the efficiency of this process was so high that we really thought it might even be high enough to really have a systemic effect on the whole animal. And to test that, we actually injected the cells into an animal model of Hirschsprung disease, where you have systemically, again, a very severe form of basically problem of an aganglionosis. And so these are animals, again, that are injected, the so-called EDNRB mutant mice, spotted little version of it. And those mice are, again, pretty severe in their phenotype, so they die typically within about 3 to 6 weeks. And what you can appreciate is once we grafted those cells, in those animals we got a pretty much complete rescue, actually, that every single animal survived that got those cells. Now we're still working more on the mechanism because, again, it was nearly too good to be true to actually be fully functional, integrated, and having full peristaltic movements within such a short period of time. And so we think, at least at early stages, we might also affect barrier function of the colon. But over time, in fact, these cells can functionally integrate and at least partially control peristalsis. Now, the other part, again, of the system is that we can use them for disease modeling. And I mentioned that we introduced various CRISPR mutations. What I show you here is 4 independent lines where we introduce basically EDNRP null alleles. And you can then very easily measure the impact of that in a migration assay. Normally, these cells migrate quite effectively. If you make, for example, a scratch, within 24 hours, it's pretty effectively repopulated. But this doesn't happen in the mutant cells. So this is such a simple assay that you can now quite easily use that for a screen. And so we screened an FDA-approved library and a bunch of different libraries, actually, and had several hits. I show you here just a few, or actually just one primarily, which is pepstatin, which is kind of a peptidase inhibitor. And it had a very robust effect. If you look how high the migration effect is, you get pretty much back to where we were before. So it's a near complete rescue of the migration defect. Now the question obviously is how this compound works. We think it acts at least in part by acting in a base-like manner. So we can take a base-2 inhibitor in this case and get a very similar effect. And we can mimic the effect genetically by knocking down base-2. Now obviously the big question now is what is really modulated by base-2, and we have some evidence that this might be neuregulin or neuregulin-related factors, but more work needs to be done on that. But what's another, I think, interesting proof of concept in this model is I showed you a little bit of cell therapy and drug screening, and so the question was, can you actually combine those? So the idea there would be that if you have a child that has a genetic form of the disease, and you have relatively quickly, in acute sense, have to treat that child, so it would be maybe difficult. In many cases, we don't even know what the mutation is in those kids. It would be difficult to fix the mutation. So what if you could just simply make matched cells, such as iPS cells, and then just simply give those cells a boost in their migratory capacity by pre-treating them? And that's what we did here. So it's basically just 72 hours of pre-treatment with pepstatin A, and then injecting them back into this migration assay. And what you can see is that normally, indeed, EDNRP migrates much more poorly than wild type. And just simply 72 hours of pre-treatment gives you a pretty good rescue of that migratory capacity, saying that at least in principle, this might work. Now what we are doing now is obviously to see, does it work for many more mutations? This is for EDNRP, and we have some evidence that it might work for RET mutation, but we also would like to see it in patients where we don't even know the mutation. And so for this story, really what we have now in our hands is for the first time a system to study the enteric nervous system. That's kind of the fundamental part, because it's a very important, very understudied part of the nervous system that's now accessible in the human system. That this does have quite remarkable migration capacity was also quite a gratifying feature, because that was clearly not the case in previous attempts of other cell types to be used, for example, in Hirschsprung disease. We also showed therapeutic that these cells seem to be able to rescue a model of Hirschsprung disease, and they can also be used as a tool to really used for drug discovery in a disease modeling effort, and again, you can even combine the two. So this leads me then now to the last part, which is exactly the part, can we take things really towards translation, and where we have gone the furthest with that is in the context of Parkinson's disease. And you've already heard a beautiful talk from Jun Takahashi, so you're already well primed for basically some of the questions that are going to come up. And so again, Parkinson's is obviously a disease that has a clear need for additional therapies. And the best used drug even today has been around for more than 50 years, and most therapies still circle around that. And as Jun mentioned, this drug still needs you to have actually dopamine neurons around to work effectively, and thereby works less and less effectively over time, because dopamine neurons die as the key feature of Parkinson's disease. And so it's clearly quite simple rationale that if those cells die at the time of diagnosis, they're already 50% or more are lost, can we just simply replace them? And so again, it's a bit the agnostic way. You don't really know exactly why the system breaks down. We clearly need to do much more work on that. But can we even just simply put a new system in? If you think of a car where the engine breaks down, it can break it down for many, many reasons, but you can just simply put in a new engine. And so that's kind of the rationale of that. And again, the rationale that has been around for a very, very long time. And fetal grafts were actually quite instrumental, and even today, in guiding those efforts, because they have shown that you can isolate fetal dopamine neurons that can engraft in patients. More than 300 patients have been grafted worldwide, and they can secrete dopamine. Now, the question is, how good did fetal cells really work? And that's a whole story by itself. We could talk for a whole hour about that. But if you just kind of do a meta-study, and that's work from Roger Parker that tried to put that together, in general, they seem to work somewhat because they improve this Parkinson's score, UPDRS, quite effectively. But the main problem has been that 2 placebo-controlled trials that were done about 10 years ago or longer than that, they basically failed the primary endpoints. And again, there's many, many reasons why that might be. Most importantly, probably patient selection. But they clearly set the field back because not only did they miss the primary endpoint, but they actually saw certain side effects which could often induce dyskinesia. And so for a long time, again, this field was kind of dead. Now, it actually became partly revived, not only from the stem cell side, but from the fetal side. Because the patients actually were followed even after 10, 15, 20 years. And it was noticed at least some patients, maybe about a quarter of a third of those, seem to do actually remarkably well, meaning they are now 15 or 20 years out. They actually are off L-DOPA medication. And if you do PET imaging, you find that they have levels of dopamine that are comparable within the normal range. So that really doesn't happen in Parkinson's patients and suggests that this can work, but obviously, how do we make it work more reliably? And actually, Roger went back and tries to figure that one out in fetal tissue, and it's clearly very valuable, and I think this work will help us again with regard to guiding patient selection for our stem cell-based efforts. But on the other hand, I think we also need to all acknowledge that fetal tissue has so many other problems that it's never going to be a routine therapy for patients. And so again, that led then on a long journey, which I'm not going to take you through, to find actually the right protocol to make those cells. And a key step in our side was really to understand that the cells have to go through certain steps using both dual SMAD inhibition, but using fluoride-derived midbrain dopamine neuron specification, like June mentioned today, which is actually a very similar model that we've described in 2011. And so this is a quite robust protocol where we showed that you make cells that don't just simply look nice in a dish, but they engraft in vivo, in mouse, in rat, or in monkey brains. And we have done since quite a bit of additional monkey studies to show that the cells really have morphologies that are very similar to endogenous dopamine neurons, and that you can do that at a scale that's comparable to what has been seen in the best fetal grafts. So typically when fetal patients, some of those patients that did really well, came to autopsy, the highest number that we're seeing is roughly around 100,000 dopamine neurons on each side. And so we could easily achieve that, in this case, in a monkey using human ES-derived cells. Now again, we did over the years many, many different assays, and the most common ones, routine assays. I'm sure you've seen all of those, are these kind of rotation assays. You don't have to worry actually about these mice. This is a sped up movie. So it is they are on amphetamine, but amphetamine doesn't work that well. So but you can basically show obviously that this rotation behavior is improved. You can show many other assays, for example, where you test basically the forelimb use in this assay. You see after transplantation you can use the left paw much better than before. And so there's a whole bunch of assays that are improved with such grafted human ES-derived cells. Now one question again that we also addressed is the mechanism of such improvement. Because there's always the question when people talk about cells, some people think even maybe mesenchymal stem cells can improve some of those animal models, and I would definitely take issue with that. If you look very carefully at these studies, you can figure out why they don't work and when they have only a very partial effect. But we need to set our standards very high too and show, do our cells really work via integration and release of functional dopamine? And the way you can do that is, again, optogenetics. In this case, not by putting in a switch that activates the neurons, but just the opposite. So you put in a switch where we can shut off the neurons. And to test that, we used yet another behavioral assays, just shown here. So you have a mouse that walks in this corridor. The mouse is hungry and it's going to eat food pellets on either side. And so what they now do is, you see basically in a wild-type animal, clearly does it at 50-50. But in a Parkinsonian animal, it's very disabled, meaning it actually ignores food pellets on the contralateral side of the lesion. Neurologically, that's considered like a hemi-neglect. It's not that it could not pick them up, but it ignores them. Now, if you graft those animals, the gratifying thing again is you can completely rescue that to 50-50. And here, to show you some controls, for example, we now have a control dopamine neuron that has a switch, but the switch is not an optogenetic switch, but just simply GFP is activated by light. nothing happens. But if you then actually do that in the context of truly switching off the graft, so you have an animal that over 4 to 5 months gets back from here to here, completely recovered, you can show that within a minute of switching off the cells, the animal is again as lesion as it was 5 months ago. And that happens within a minute, meaning that indeed it's the activity of the graft that causes the loss of function. And you can show pharmacologically that this is actually dopamine-dependent activity. And so therefore, what we showed in this study is that what you do have to have for functional improvement is activity-mediated dopamine release. But it didn't really prove yet that it's releasing dopamine at the right place. For that, you need to do one more step that was again alluded to a little bit by June in the morning when he tried to trace the cells and show that they hit the medium spiny neurons. So here we also look at the medium spiny neurons, but we actually look at them functionally. So they are marked here in blue and red, and we electrophysiologically now record from those neurons that normally get input from the cortex or from the thalamus coming through the corpus callosum. And so now what the dopamine neurons normally are supposed to do is to modulate the strength of that synapse. Dopamine is a neuromodulator. And so what you can now do is, in fact, show that's exactly what our grafts do. When you now measure, again, either from D1 or D2, red or blue neurons, you can show as soon as you switch on the light, you shut off the dopaminergic input to that synapse, and you see the EPSP drops by about 40%. If you then switch it off, it comes back to control. And so that shows indeed that the cells modulate that input. And we even then went further, I don't have time into that, pharmacologically show that this specific effect was actually mostly mediated by the D1 medium spiny neurons. But coming now back again to the actual translation, so how do you now take this evidence of having something that seems to be quite interesting into the clinic? And so we are fortunate again that we got money from the state to really try to go on this road, to really try to get to the clinical trial, hopefully. And so we actually decided to go with a human ES product, and that was mostly due to regulatory reasons, that in the US, again, several trials of ES-based products have already been approved. So far, no IPS product. And if you don't go matched, we don't really see a big advantage of IPS cells. And so we decided that already somewhere in 2014. We already, at that stage, made a very large bank of ES cells together with Weissman that is basically fully tested for the various assays you need to do for making the FDA happy with regard to specification of those cells. Now, a key feature was then to take that into a GMP-compliant protocol from the protocol that Sonja Krix developed many years back in the Nature paper, and that actually turned out to be a little bit challenging. So when we started using GMP reagent, there was kind of a protocol drift, and this shows you just a bunch of qPCR runs that we routinely run. We run, for every differentiation, 48 different markers, and some of them started to wiggle, not the ones that we usually were concerned of, like FOXA2 LMX1A, which were some of the key features of making dopamine neurons based on our Nature paper. But some other markers like PAC6, this is actually CT threshold, so low levels are basically higher expression. So we saw suddenly more PAC6, which shouldn't be there, and we saw lower levels of TH. And so we then basically went forward and started developing optimized protocols, had to go back to developmental biology basically to get something that's not only getting us back to where we were, but actually make certain other markers that were relatively lowly expressed in the Krix et al. Protocol much, much higher expressed. To just give you a little bit of a flavor of this protocol, which actually still unpublished, even though we've used it now for quite a while, it has to do with Wnt signaling. And so it was actually kind of an interesting little controversy when our paper came out and subsequent papers with regard to the concentration of the GSK3 inhibitor that you use. So we use typically 3 micromolar, and June actually does the same thing, but there were other groups that showed that they get the best result with 0.7. And so what this Wnt inhibitor, this Wnt activator is supposed to do, it caudalizes in a dose-dependent manner the cells to the midbrain hindbrain region. And so we actually found out that the difference between the different protocols was just simply KSR. So it turns out if your medium contains KSR, KSR contains component, which are actually, there was a paper by Roel Nusse later on that showed that the KSR contains, I think it was an arachidonic acid-like factor that inhibits Wnt signaling. So that explained the difference between the paper. That's great. But so we then, basically, when you go to a GMP-compliant medium, you go more to that level. The problem was, in all those protocols, it's actually quite a wiggly protocol, so meaning sometimes the optimum is at 0.7, sometimes at 1, sometimes at 0.6. And that makes the protocol not very scalable. And so what we found out is actually that to solve that, you need to have a second wave of Wnt signaling that we think has to do with the fact, yes, you need to suppress the forebrain early, but then you have much higher levels of Wnt that basically is expressed at the midbrain organizer. And so we can mimic that in our conditions quite effectively, using, again, a pulse of Wnt at later stages that now quite massively induces, for example, engrailed, much, much higher than what you get with the standard protocols that is used in the field. And that high Wnt level then leads to certain markers highly expressed that were, again, often not seen in the field, much as PITX3, that are now part of our release criteria for our cells. Now, mechanistically, the way that seems to work is that this protocol is really highly dependent on Wnt-dependent engrailed expression, because you can now make this modified protocol where you get, again, nice engrailed expression, but have an engrailed knockout here done by CRISPR-Cas9 targeting. And if you do that, you actually see that engrailed is essential, for example, to suppress certain genes like PAC6, and again, to allow PITX3 expression, and also to suppress other markers such as a so-called STN marker, subthalamic nucleus marker, that is also very closely developmentally related to dopamine neurons. So we think, again, this protocol is really quite powerful to give us much more robust data. And we had to show that, and again, now the last 2 years in 35 independent runs to show that you can do that routinely again and again and again to get a product that's very robust and very pure. And so that's, for example, based on certain marker like FOXA2. That's the marker of the floor plate versus PAC6, which are other neuroepithelial cells. And you see that you get about 99% purity of getting just the floor plate versus neuroepithelial cells. Now, the next step is then making these cells really in a GMP facility at scale. And we are fortunate that at Sloan Kettering, we actually have on-site a brand new facility that has up to 16 different suites. And we basically were occupying the last few months those 2 suites quite extensively and started producing the cells after, again, 2 years of optimization at large scale. And so we trained, again, our people to actually be in this facility and are now making, again, the actual lots. Not only, again, test runs anymore, but the actual lots that we want to use in patients. So meaning we made 4 large lots, typically between 1 or 3 billion cells now, that would correspond to the equivalent of about 1,000 human doses. And so those lots are now the ones from which we now have the luxury or the duty, I guess, to figure out which one we're actually going to use. So we have to, for the FDA, need to basically show reproducibility that you can make the process. And so now we could show actually that in every single case we met the specification that we set ourselves, but now we actually have to decide what's really better because they all met the specification. And so what you then have is a little bit like a specification sheet, a little bit like on a cereal box where you can read all the ingredients. You have now all the ingredients of what's in your box or in your vials. And so it has all these markers of TH and PITX3 and so on to meet the specification that are now hopefully ready to move forward. And so a key feature also in this whole process, if you make 3 billion cells, you don't want to do that for every single patient. So we want to have an off-the-shelf cryopreserved product. And so what we were able to indeed optimize those freezing conditions, where we now have conditions where you have about close to 90% viability when you thaw your cells. And not only 90% viability just when you thaw them, but even up to 24 hours after you thaw them. The reason why that's important is if you think of how the cells are going to be used in surgery, the surgery might take several hours. And to get the cells to the surgeon might take some time too. So you want to have enough time to have a truly stable product. Now, the other component for off-the-shelf therapy is the true shelf lifetime of your cells. And that's a little bit of a challenge because, again, it's obviously something you can only test over time. So but right now we have solid data for 12 months, and we're actually just getting to the 18 months time point where we seem to have, again, good stability of that product over time. Because, again, we already made the cells that we want to use in the patients right now, so they better have to be stable during the whole trial phase. Now, also equally, or maybe even more important, is that the cells, after all these things, are not only viable here in the dish, but they're viable in vivo. And that's what you showed here, that indeed you can now graft these cells back into, this case, a nude rat. And you showed not only that the cells survive quite nicely, but they have this very extensive fiber network that they extend, which is, again, a key criteria of a dopaminergic therapy, because you need to reinnervate the whole putamen at least. We also see that the performance of the cell functionally is as expected. Exactly at the time when they are mature enough in vivo, they start leading to a complete recovery of that rotation assay that I showed you before. Finally, we did also some pilot studies in monkeys again, just with the final frozen product, to show that the cells also in a monkey can survive and basically continue to express TH. Now, the remaining part is really now the fact that you need to go through the hoops, or jump through the hoops that the FDA puts up. And one of those hoops that actually you can argue again how good of a hoop is something which they call is basically spiking studies. So the idea is obviously you work from a pluripotent stem cell line to start off. Pluripotent cells could form teratomas, and we heard again from June quite a bit of the risk of teratoma formation. And so we are pretty confident that we can no longer detect any pluripotent cells, but we need to show to the FDA that because any assay has a certain level of detection, is to what level of contamination would you actually see a problem. So you start mixing in either 100% ES cells, 10%, 1%, 0.1%, and you see in which animal would you still see teratomas. And we could see that when we have up to 1% undifferentiated ES cells, we can see teratomas. Obviously in our product we see none. And if you have 0.1% contamination, we also see none. And again, it's a little bit useless as an assay for detecting, again, how many pluripotent cells you have, because there are much more sensitive assays, both basically qPCR-based or by flow, we can at least 100 times more sensitive than that assay. But again, the FDA still insists for us that we need to do this in vivo assays, because the argument is obviously, here we do just a few animals, we did 6 animals. What if we did 50 animals, 100 animals? Would you see something? That's kind of their argument. But again, that leads us now to the kind of some of the things we still have to do, which is we had, again, the pre-IND meeting where we basically more or less agreed to the final studies, which are basically 3 types of safety studies. One is the tumorigenicity study, toxicity study, and a biodistribution study, which are done in nude mice, and actually in NSG mice, which is the most immunocompromised strain. And so those include, again, for example, the spiking studies. Toxicity studies are similar to drug treatment, where you have to look for liver toxicity or cardiac toxicity. Clearly, we don't expect that from cells staying in the brain, but you have to do it. And biodistribution goes to this question that, in fact, they do stay there. We also do another set of efficacy studies with the final product in about 40 animals. This is then done, actually, in this case, in nude rats. What we are actually quite happy about, it looks like we at least need to do no efficacy studies in primates, which makes things more supportable and therefore less expensive. And so we are actually, again, we did quite a bit of primate studies before, but we don't need to do that on the final product again. And so again, this study should be complete in 2017, and then allow us to indeed go to the clinical trial. And that's then basically the last step, obviously. What is the patient population to really getting those cells? And so we think obviously it has, by definition, to be a phase 1 study with the goal of safety and tolerability. And we clearly, at the same time, with all the experience of fetal grafts, we clearly have to look for early signs of efficacy and biomarkers, which you can do both by clinical rating scale, a whole bunch of classic scales. We do it also by using more advanced techniques like Android-based devices. There's a whole bunch of new techniques that hopefully are going to be more sensitive to see as early signs of efficacy in those patients. But then also PET imaging is very important, because that actually seems to come on based on the fetal study slightly earlier than the clinical improvement. We're going to graft basically 10 patients with, again, certain inclusion-exclusion criteria, which I'm not going to go into great detail here. And we're going to plan to have 2 doses. This thing, I think, died. We're going to have 2 doses of, let's see if this works. We're going to have 2 doses of cells. In one dose, we try to aim for the 50,000 surviving dopamine cells, and in one, we aim for the 150,000. If you remember what I told you before, is that the 100,000 was the higher end of the patients that did extremely well. So actually, some patients had only 30,000. They also did well. And so we kind of chose that as a compromise for a first-in-human trial, because obviously you don't want to overshoot either. And we think that's a reasonable compromise to move forward. Then, because we have an ES-based product, we decided that we want to have this patient immunosuppressed for the initial 12 months. And that's again a little bit of a controversial issue that June again alluded to, that actually for allografts, grafts do not get rejected. And in fact, there were fetal grafting patients that were getting no immunosuppression at all, ever. And they had 15 years later pretty healthy grafts. Now, again, there are force and back why you still might want to immune suppress. And again, June mentioned some of those. You get some microglial infiltration. You might have slightly fewer dopamine neurons survive. And I think, again, for the first-in trial, we don't really want to take an additional risk by not immunosuppressing. You want to have the cells clearly survive. But in the future, it will be very, very interesting to see whether you absolutely need immunosuppression, and if so, whether you again want to have patient-matched cells, universal cell lines, or any other strategy to move forward. Then last but not least is obviously the surgical procedure. So we're going to use, as you would expect, stereotactic surgery. So these cells have to be injected directly into the brain, primarily in the region called the post-commissural putamen. Now, for the initial trial, we actually do that in a little bit fancy way where we use it under MRI guidance. So this is basically an intraoperative MRI where you can very effectively target, like with any stereotactic system, but you can literally watch it happen. So this is shown, for example, for the same device that you want to use, injecting, in this case, a drug into the striatum. And you can see that you can actually follow the injection of this compound in the specific site. Now, we don't really think that's the limiting factor, that you would not properly target it, but the main rationale for using MRI guidance in those initial trials is the fact that if you do that, you have additional safety. And the safety comes from the fact that we want to have in one session grafting both sides of the patient's brain. And that's something which the FDA is actually, again, somewhat worried. And again, we have still some negotiation with them to figure out where we need to do that. From the beginning, we can do bilateral, which we would like to do. Because for the patients to have two surgeries in different time points, I think is not ideal. And you could even imagine, again, from the immunological side, it's not good to first have cells in and then the same cells going in again. But again, you obviously need to be able to show that you can do that safely. Because what the surgeons tell you, the official thing they have to tell the patients, every needle track you do in the brain is a 1% risk of some bleeding. Most of that bleeding is completely harmless, but you have to obviously be aware that it could be at some point not harmless. And if it were not harmless, I think we could easily pick it up in MRI and therefore not do the second side. And so I think, again, that is an additional level of safety that for us, I think, is quite important to really move that forward in an efficacious way. Then last but not least, I want to make the point again that we really try to do that not in a vacuum, but we try to do that in a way where we discuss quite a lot how to move that forward most effectively. Because as we know, both from the gene therapy field and even from the Parkinson's field with those placebo-controlled trials, I think they were maybe done at a stage where we didn't really know enough how to make a placebo-controlled trial the right way. And so we discussed that again with various groups that actually want to move forward clinically, both the group we heard from Jun Takahashi, but also Malin Parmar and Roger Parker in Europe, and obviously our group in New York. And I'm sure there are going to be other groups that hopefully going to benefit from this experience and making that a safe effort. And we heard, I think, from Jenny Loring again, that not all people really prescribe exactly the same way. We have to deal with groups, for them again, the trial that's going on right now in Australia that doesn't really follow the procedures that we think are the right ones to move forward, and where we are again quite worried that those trials could actually make our life much more difficult in the future and make it much more difficult to make this therapy coming to fruition after, again, nearly 20 years of trying to develop it. And last but not least, I want to acknowledge the people that were really instrumental for what I showed you today. In the first part, I showed you the work on familial dysautonomia, which is currently just been carried out by one postdoc, which is Nadia Zeltner. And the dendritic neuron work was done mostly by a very, very talented graduate student, Faranak Fatahi. And the Parkinson's work, again, involves many people. I cannot really mention all of them. I showed just some of the key people. I need to particularly mention Mark Tomishima, did an amazing job now in getting those clinical grade batches ready to roll, hopefully. Stefan Irion, who is our program manager, and Vivian, who is the co-PI of the project and also the surgeon of the program. And so again, it needs a whole team really to make that work, and hopefully again, we're going to get it to work and going to move towards the clinic very, very quickly. And again, this would also not have been possible without funding, and there I want to particularly mention the New York State initiative, which is NICEDEM, not to be confused with NICEF, which is not a great organization, but it's actually a state program, not NICEF, that really made that possible and has a really very instrumental role, similar to CERM, to really bring some of those efforts now, hopefully, to the clinic. And I'm going to stop here, and I would be happy to take any of your questions.
Great talk. Questions for Lorenz? Sure.
I'm thinking of it a second.
So, Lorenz, I'm just, one thing, I'm afraid I probably had a brain glitch because I didn't understand whether the FDA was going to allow you to do bilateral transplants or not.
So, I mean, we had basically in the formal meeting, we had a lot of discussion on that issue. Yeah. And we kind of convinced them that it might be the better way to go, but we need to justify it. And there is still some argument that maybe the first or second patient out of the 10, we might have to do unilateral. But that's still something which is kind of in negotiation. And as you know, you don't really get a complete answer. I mean, at the end, it's the IND where that gives you the final answer. You can make your point, but you have to make that point again with sufficient data when you actually submit your IND.
We appreciate you making those points. Thanks.
My question is the midbrain dopamine neuron you made from EA cells. Before transplantation, I'm not sure you made a progenitor cell. After you transplant into brain, they turn into a dopaminergic neuron, or you already made a midbrain dopamine neuron before transplantation. They survived. So the image you show in monkey. That's question number one. Question number two is there are many multiple group has generated a dopaminergic neuron from EA cell or iPS cells. I heard your cells, they were called true DA neuron, dopaminergic neuron. So what make your DA neuron better than anybody, another group cell generate?
So for the first part, if I understood you correctly, I mean, it was basically the stage of the cells where they're already dopaminergic or they become dopaminergic in vivo. So they are just kind of, it's like the Goldilocks thing. So they are not really, obviously full dopamine neuron for sure, but they're also no longer progenitors. They're just basically in the last division to become a post-mitotic dopamine neuron at the time when we graft them. And so that we found just empirically to be a good stage where we can both freeze the cells, saw them successfully, and graft them, have good fiber outgrowth. And so you probably could go a little bit later, you could probably go a little bit earlier. There's some wiggling room, and some of the different groups actually go at a slightly different stage. But in either case, for the cells to be fully functional, if you looked carefully, our assays took 3 to 4 to 5 months before they actually recovered the animal. In the human, it might take even slightly longer, and that's because they continue maturing. The dopamine transport or other dopamine machinery continues to mature over that time, which is the normal developmental scale of those cells. So they definitely continue to mature in vivo. Now, with regard to the different protocols, it's obviously a tricky question with regard to how do you know you have the right cell? And there's still, even today, again, I mean, I showed you this tweak to our protocol with the boost of Wnt signaling that we still think makes a difference. It doesn't mean that you cannot get the right cells without that boost, but you get a much lower frequency. And so then the other issue is that within the midbrain, there are also different populations from anterior to posterior. And I think it's actually a paper going to come out from Thomas Perman and others that show that you can very easily confuse FOXA2-LMX derived cells with a region called the subthalamic nucleus, more anterior. And so in some cases, actually, people get contamination of those most anterior cells in the mesodencephalic region. And so I think this new protocol is going to resolve. You completely get rid of those, and you get also rid of the more posterior ones. But so again, the stringency continues to increase and increase from the initial papers we published in 2004 to the ones in 2011 to the ones we haven't even published yet. So I think the more we learn, the better you understand it. And again, the question is, how does that match the endogenous neuron, and what's really the right population? And even now, no one has really truly completely a pure A9 population. And so I think that's still something that needs to be further resolved.
I'm wondering, do you think the age of the niche that you're implanting the cells into can be a factor how would this affect the population that you're going to treat?
No, it's an interesting question. And again, we kind of rely a little bit, again, on the fetal grafts, because obviously those were done in patients up to 60, 70 years of age. And we did some studies where we grafted them into aged monkeys. And we didn't really see a huge difference, but I don't think it's completely negligible either. I'm sure there's some differences, but I think it's good enough that you should even get in an aged brain, basically it starts to survive and hook up. The bigger problem is if you wait too long in the disease, because if you wait too long in the disease, you get degeneration of the cells that actually receive the dopamine. And so then obviously it's not going to work because the patients don't respond to dopamine in general.
Do you know how mature your cells are compared to like the fetal cells? Because there are differences from embryo to fetal.
I mean, once they function in vivo, presumably they are comparable. But when we graft them, they are probably, I mean, again, fetal cells is also the problem, depends which age of the fetus they chose. So they chose ages typically from up to 5.5 to maybe 9.5 weeks post-conception. So ours would be more on the early stage. They'd be more like maybe 6 weeks, comparably.
Okay.
I would like to close with one more question.
Sure.
Going back to the disease modeling, the first story that you told us, some of the variability seems to come from the second hit in a different gene. Mm-hmm. And I must admit, I haven't read that paper, but did you try to correct or to stimulate that mutation artificially to see if that gene is causal?
No, that's clearly something we are testing now, which of those mutations really can do that. And again, so far, we don't really have evidence that one of those is completely enough. And so, again, we don't really have a nice answer, but clearly we have candidates to be tested. And I mentioned very briefly, we did, again, also a drug screen where we also seem to be able to overcome it indirectly. And so we kind of tried to, from two different ways, both genetically and kind of chemically, try to see where we see a convergence in those pathways.
All right, so I'm sure we can continue the discussion over the reception outside. So let's thank one more time Dr. Lorenz.